Hello Achievers, welcome to Team MDS Achievers, myself Dr. Vikram. Today we are here for the discussion on the topic of biochemical reactions, that is transcription and translation. And in the next chapters, the videos, we will be discussing about the Krebs cycle and glycolysis also. See, firstly, you should be knowing that few terminologies, that is like, see, why we are studying this concept is first thing we should be knowing before we move on to the topic. That is like, generally after we do any kind of, that is like implant surgery or any kind of like surgical extraction of any teeth or also root canal therapy, then what we'll be doing is we'll be prescribing antibiotics, right? See, we should be knowing that the mechanism of action of these antibiotics, because some of the antibiotics will be go and inhibit this transcription and translation process of this bacteria or any microorganisms, right? So thereby, as a doctor, we should be know that this transcription and translation process, so that we can as a doctors we should know that, otherwise we will be like an RMP doctors prescribing the antibiotics only, right? So let us move on to the transcription and translation. Firstly, transcription is a process where the mRNA is synthesized from the DNA, and translation is a process where the proteins molecules are synthesized from this mRNA, which are the protein molecules are the building blocks of the, right? So thereby this process occurs in a chain reaction, that is like one after other. So we will be doing starting from the transcription, that is like, suppose let us start from the basic concept, that is like, see, every organism is made up of as a human being we are made up of organ systems are made up of organs are made up of tissues are made up of cells. In that one particular cell where the biochemical reaction is started and also occurring, that is like, in that biochemical reaction of the cell, the nucleus is present, right? In the nucleus the DNA is present, where the protein synthesis, mRNA synthesis starts. Once there is a need for the synthesis of protein, there is an action of the transcriptase enzyme on the DNA, which causes these helical structures to open, uncoil it. Then what happens is that there is a laying of this chain initiation starts, and the transcription occurs in three phases, that is like initiation, chain elongation and chain termination are the three steps that are involved in the transcription process. And the initiation occurs in the presence of a transcriptase enzyme, and also sigma factor is a factor that is required in this chain initiation. Okay, then moving on, that is like, once this transcriptase enzyme and also sigma factor are present, then it uncoils, then the mRNA is being laid on from the one prime end to another prime end, and once then chain reaction is started. Then chain elongations, we see ATGC, that is adenine, thymine, guanine, cytosine are present, right? So what happens, these are laid on it on a sequence manner, okay, accordingly, like in an antagonist and agonist manner they will be laid on it. And once it reaches the chain termination where the codon reaches like a termination codon, then the mRNA molecule that has been prepared will be terminated and it will be secreted out from the nucleus. That is, it exits out from the nucleus and enters into cytoplasm. See, from here onwards the translation process starts. Okay, basically the translation process is like synthesis of protein molecules, we know, right? In this you have to know that mRNA is, which meaning that messenger RNA, it just carries a message from the DNA to the ribosomes, which are the protein factories, right? And the tRNA, which is called as transport RNA. Okay, this, once the mRNA reaches into the cytoplasm, it goes and attaches to the two molecules, that is AMP and enzyme. Once they are attached to the mRNA, it results in the activation of the mRNA. Okay, once the mRNA that is activated, then this activated mRNA goes and binds to the 3' end of tRNA. This is very much important. See, 3' end, which is a card, that is like COOH group is present on the 3' end of the, one of the very, why I am saying this is very important is like, this was a question that was asked in the my time, that is like a NEET MDS 20 time. Where does the mRNA go and attach to the tRNA? Is what the question that they asked. It is the 3' end, which is a carboxylic oxygen group is present, right? Once it is attaches to this tRNA, then this adenylate complex, that is like, see, AMP and the enzyme that are activated the mRNA will be saying tata bye bye, my role has been completed, and it will go out. and it will find for another mRNA in the cytoplasm that are present. See what happens once the tRNA is attached to the mRNA, which is chain reaction, right? See, tRNA has a 3' end, 5' end and a T-size loop. So we are not concerned about the other loop, other parts, as it is mRNA is attaching to the 3' end. Okay. See, each and every part has its own action also. See, once the mRNA is attaching to the 3' end of the tRNA, it will transport the mRNA from the cytoplasm to the ribosomes. That is, smallest ribosome it will be taking. There are two types of ribosomes, okay, right? Smaller ribosomes and larger ribosomes. It will be taking to the smaller ribosome, that is 30S or 40S type of ribosomes. And once it takes this mRNA to the smaller type of ribosomes, then it will be start creating a reaction where the larger ribosome comes, that is like either it is a 50S or a 60S type of ribosome, that is based on the prokaryotes or eukaryotes, that is like 50S or 40S, 60S, sorry. That is like if it is 30S, then 50S ribosome will be attaching to it. If it is a 40S type of smaller ribosome, then the 60S type of ribosome will be attaching to the smaller ribosome. Then once these ribosomes are attached to each other, then the protein molecule will be started. That is like, suppose this is mRNA like this, a chain molecule, then the smaller ribosome and larger ribosome will be start coding from it one end to the another end. Okay, once it starts coding on it, the transport RNA will be transporting it on reading in a manner like a system, right to left and left to right, like that. From the, as we read in the three, right to left or left to right, but whereas in the molecule in the cytoplasm on the mRNA, it will be from the 3' end to 5' end. The chain reaction will be continued. Okay, once it continues and forms a protein molecule at the distal end of this or 3' end of the tRNA, then once the coding of the mRNA is completed, then the protein molecule is released out from the tRNA and the ribosomes will be separating. See, as it has an mRNA, they will be separating. Once the code reading has been completed, then they will be separated apart and thereby they will be released in the cytoplasm, okay, to carry out another functions. And thereby tRNA, which is attached to the protein molecule, also will be terminated and tRNA will go and find for the another mRNA that is present in cytoplasm. And this protein molecule that has been synthesized, right, it goes and exhibits its own activity. This is how the activity of the transcription and translation process occurs. Okay, see, these are the concepts that you should be knowing as a doctor. Like, it's not just for the sake of marks, but it's also about like subject. And also why we do, you might have a doubt that why you have to choose the MDS Achievers Academy, right? See, the one thing that we are concerned is to bridge the gap between the clinical practices and theoretical knowledge. See, the one who knows the theoretical knowledge and also who knows implying it in the clinical practices are the ones who are successful in the clinical practices, right? And the major basis for the NEET MDS exam is also to know and also set the certain standards in the exam. That is like a person is qualifying in the NEET MDS, he or she knows certain knowledge about the clinical knowledge and also the theoretical knowledge is what they are meant to be. And they will be asking the question also in the theory, that is theoretical concepts that are implied in the clinical practice is what they are concerned about. And if we know this concept only, then what happens? We can easily crack the NEET MDS, right? See, this is the main agenda of the NEET, like MDS Achievers Academy. That is like bridging the gap between the theoretical concept and the clinical implications. Once they are clubbed together, definitely it will be success. Thank you. This is Dr. Vikram signing off.